My name is Chris Johnson. Again, I work for Dr. Neil Gudmestad up in Fargo, and we've been doing Liberibacter haplotyping work for quite a while. And now that we have a few genomes, we decided to do some comparison analysis. And our big question was, we've sequenced these genomes, we've identified sites that in our assemblies are different. And so we just kind of like quick and dirty wanted to see, are these variations that we observe in the assembled genomes consistent across our samples? So like you've just heard, we have two Liberibacter haplotypes. The published haplotype B genome that came out of Dr. Hong Lin's lab in 2011, that's the ZC1 genome that represents a haplotype B sample. Haplotype A genomes have now been sequenced from the US, insects supplied by Dr. Don Henney, and in New Zealand they have haplotype A also. We have a second sequenced haplotype B genome from Texas that was also from Don Henney, from a colony collected at Edinburgh. So the big question for us is, we have these genomes, what are we going to do with them? We can solve or answer some questions on taxonomy for Liberibacter. We're looking at genetic diversity in these populations. We can use genetic diversity to try to predict some epidemiology. How's the Liberibacter moving? How long have they been here? Which ones are new? Which ones are old? We'd like to do some population biology and different plant hosts and different insect vectors. And then, obviously, biology, physiology. We can track infections in different locations, different temperatures, different hosts. So the genome that I've been working on is the LSO USA1 genome. Right now we have it in 7 contigs, and there's the contig sizes are listed there. The bulk of the genome is represented in the 686. We're having problems placing the phage. We know there's two phage in our genome. Much like the ZC1 genome, the phage are the identity between the phage is very high. And there's also up to 9 phage remnants throughout the genome. So placing exactly where the genome or where the phage are in the genome is difficult. We ended up getting some PacBio sequencing for the New Zealand genome, which really helped solve some of those long reads. We haven't submitted anything from the US yet for PacBio or getting any longer reads, but we will have phage contigs to add to the sequence data on NCBI after it gets released. The big takeaway is that the core genomes between the two Liberibacter strains, between A and B, are roughly 98% identical. So when you're looking outside of the phage, when you're looking outside of the phage remnants, they are very similar. So this is another Moave alignment. I won't go into mine too much. Rebecca's is actually much prettier. She actually ordered the contigs so that they matched up better than I just threw mine up there. But the big takeaway is, yeah, you have the bacteriophage, and look at, there's little scattering remnants of phage. That the top is LSO USA1, and this is ZC1. So you can see the phage in ZC1, there's little bits and pieces of it scattered throughout the genome, which makes assembling what is truly a phage contig and what is truly a phage remnant contig incredibly tedious, and which is why I haven't finished it yet. So basically what we did is we took our two reference genomes and we mapped our reads back. That was the wonderful thing about having the sequence data from the Edinburgh colony. And so we ended up we would take one genome as a reference. Oops, back, wrong button. We would take one genome as a reference and map the reads from the other one back and look for gaps. So these gap areas, so what you've got here is a reference genome. So this is the I think for this particular one, this is the haplotype B genome, and these are reads from an A sample mapping back to it. So you can see in the B genome at this particular region, there are no sequences from A that map to it. So this is identified as a unique region in the B genome compared to the reads that we have from our sample A. And so we decided to identify several of these regions, and we were going to use them for haplotyping. So we'd pull these regions and just run a blast back to NCBI to make sure we're not hitting any other multi-copy elements or elements that are found in any other bacteria. And then we would run standalone blast against our own source reads, and that would tell us, does this fragment truly map to this genome when it's not coming from a Serratia or a Pseudomonas or some other contaminant. And then what we did was we designed primers, and I really only designed one or two primer sets from each of these fragments, but we still ended up with a pile of primers. We had over 40 primer sets designed and tested. We also had the New Zealand group sent us a batch of primers too that they identified through the actual coding regions that were missing. And then we just went through Neil's hundreds and hundreds and hundreds of samples that he makes us save from everything to find a good set of samples going back a few years. We had plants, we had insects, haplotype A samples, haplotype B samples, and then of course some mixed, and then we just ran the primers. This is an example of what a blast looks of these identified regions. And you can see here, this is actually reading conserved across all the Liberibacter genomes. The top one is solanacearum. The next one down is actually, it's one of the LAS genomes, and then another LAS genome, and another LAS genome. And I want to say this one is Americanus, I believe, Liberibacter americanus. So most of these regions are pretty highly conserved across Liberibacters. This looks like a unique fragment to solanacearum. But yeah, and this is a pretty good sized chunk. It's a 3400 base pair insert found in our genome, and we have no idea where it came from. I even ran a blast X on this fragment, and it gave me back absolutely nothing. So what I did is I picked a selection of primers to just kind of give you a highlight of what we were seeing. For the most part, all of these samples here are haplotype A samples. The matching blue primers are designed to amplify off of haplotype A. And this is our LSO FHLBR, or standard diagnostic detection primer. On the end here, you're going to see these are some phage primers. These are designed off of the ZC1 genome. This green one here, I designed that to differentiate the New Zealand haplotype A from our haplotype A. I only screened it in the design process against our haplotype A. I didn't screen that against haplotype B. That's going to be important in the next slide. And then this is a phage that was found, phage sequence from haplotype A and phage sequence from haplotype B. The big takeaway from all this is that if the primer is designed from A, it almost always works on all of our A samples. There are some exceptions. I have primer HA12, which for whatever reason amplifies from an unstable location. I got some samples from Nora Olson's group last year that HA12 does not amplify from. I actually went back and went through all of the KT samples, and this primer set does not amplify from any of them. So I have to talk to you guys to find out the backstory on that sample a little more maybe. We also have HA2, for whatever reason, doesn't amplify. And this is a fairly old sample that came out of South Texas. For whatever reason, it doesn't amplify. But for the most part, if you have a primer designed from A, it does amplify from A. So these are mixed samples. And now that we've been running cybertyping, we actually find that we do have a lot of mixed samples. And generally speaking, the whole reason we designed these primers was so that we could titer co-infected samples, so that if we had a co-infected sample, we could say, all right, this much of the Liberibacter's A, this much of the Liberibacter's B, so we can do ratio testing. And that actually works really well. You can see, generally speaking, if it's an A primer, the CTs are consistent, and the CTs are going to give us a relative titer. The higher the CT, the lower the titer. So you can see like a CT of 20, that's a fairly hot plant. That thing was smoking with Liberibacter. In 18s, And generally speaking, some of the samples you can actually see there's a really nice mix. These were insects actually collected by Dr. Secor in Guatemala, and they were mixed. But most of the Liberibacter in this particular sample was A. You can see that the CTs here are lower, which means they have a higher titer. But there's definitely some B mixed in there. We're consistently getting B titers that are much higher, or B CTs that are much higher. The titers are lower. So this is a really interesting or a really useful way to use these primers. Once again, though, we do have HA12 misbehaving on us. It is amplifying, but it's actually amplifying with a B characteristic CT, which tells us that, yeah, it's an A. It's supposed to be an A target, but this site is actually probably amplifying from the B genome in that particular sample because the CT is so low, which indicates that there's a high titer. And then once again, what we wanted to look at with these phage primers is that the phage terminase and the phage primase are generally considered highly conserved genes. You would expect those in a functioning phage to be highly conserved if they're functioning. Where the phage structural gene, historically phage structural genes have a pretty high mutation rate, so you would expect them to be less conserved. And these were all designed off of the ZC1 genome. And you can see that they generally follow the haplotype B titers, but we are missing the phage structural gene in some of them, which tells us that, that it does seem to follow that pattern. And the other interesting thing is our NZ1 primer set, which was designed to amplify New Zealand haplotype A versus US haplotype A, amplifies quite nicely from some of our samples at B titers. And so it does look like that priming site does occur in some of our B samples. And then you can see here on the end there's some phage primers, and they do amplify off of both. So next we have the haplotype B samples and primers. We have a really good collection of haplotype B samples. And we do have some, once again, these are all B samples. These are plants. These are insects. We do have a few of our A primers that are misbehaving. HA12, once again, is hitting a couple of our colonies. And what was odd was I actually went in and we have this colony still active. I extracted 3 batches, 2 fresh and 1 from our -80 freezer, and reran HA12. It amplified from 2 of the 3 samples. And then I actually ran it twice. So that priming site seems to come and go from some of our samples for whatever reason. I'm not quite sure. HA14 also gives us kind of an off CT on this particular sample. I tested it, it's there, but looking at the annotation, some of these regions are really poorly annotated, and they don't blast very well, so identifying exactly what's going on at some of these regions is difficult. HB1 is actually, there's a repeat right next to HB1 in the ZC genome, so part of that region is actually repeated elsewhere in the genome, and I think maybe there's a somewhat of a splice variant because HB1 doesn't amplify from some of our regions. But like I said, I had 30 haplotype A primer sets and 16 haplotype B primer sets, and I only had 2 or 3 A's that really didn't behave the way they were supposed to, and this is the only B that was giving me fits. The big takeaway here, though, is all of these primers on the back end here are phage-based. And look at the diversity. I mean, it's almost random that some of them amplify, some of them don't. And this has all been tested multiple times, and it's consistent. We have this particular sample only amplifies 1 of our phage targets out of 5. And then really the phage 1, we didn't expect that to amplify because that was designed off of our haplotype A. But even that one amplifies from some of our samples. And so looking at the haplotype B samples, although the core genome it seems to be fairly conserved, we have a lot of phage diversity in our samples. Okay, so the results. Most of our sites are consistent across a single haplotype. So if you design primers, or if I design primers off of haplotype A, most of the time they work. There's just a few exceptions, and we won't hopefully give you those primers. If I do, I'm sorry. The primers can be used to quantify individual titers in a mixed sample. So doing competitive inoculations with different strains, doing competitive transmission studies with different insect haplotypes, different host plants. We can use these primers to do all that now. The phage-based primers are most likely to amplify across haplotypes. So they're the ones that are going to cause problems. They're going to amplify where they maybe they're not supposed to. But at the same time, the sequence conservation across the phage does seem to indicate that they are important at some point somehow. The HA12 primer, like I said, I retested on several samples. Sometimes it amplifies even from the same colony. Sometimes it will amplify and sometimes it won't. We did have several haplotype A specific primers amplify from B samples. Most of them were in the phage. So there is some cross between the genomes, particularly in the phage area. And then like HA12 and HB1 do not amplify from some of their type-specific colonies. And we don't have, I don't have data yet. I don't have contigs from, on those regions from colonies that don't amplify them to see exactly what's going on, but that'll be coming. Primer NZ1, which I originally designed to amplify only from New Zealand, because I didn't screen it against the haplotype B samples, I was surprised when it did hit several haplotype B samples. Then I actually went back and designed another primer set called NZ2, which I screened against our haplotype A reads and our haplotype B reads. And I've run that on over 150 samples from the US, from Guatemala, from Honduras, and from Mexico. I have not found a single US sample that primer set amplifies from, or Central America. But it does amplify from, I think I have 5 or 6 samples from New Zealand, and it does amplify from all of those. I had Sarah Thompson back in New Zealand test it on some of her samples, and it does amplify from hers. So it does appear to be a valid primer, but for whatever reason, it hasn't amplified off of anything from North America or Central America yet. So our discussion, QCR primers work to quantify individual targets. Some of our sites are unstable, which these sites might be unstable. In our haplotype B assembly, we actually look at the phage insertion points because we have really good phage structure, and we can see that in at some of those loci they have phage or they don't. So you can see where it will read out of the core genome and into the phage, or it will read straight across the core genome and skip the phage entirely. So our big question is, do we have a mix of Liberibacter populations or genomes, or do we actually have the phage actively moving in and out at those locations? And so that will be tested here in the future. We'd like to develop a better phage model, and that will come when we get better phage contigs, particularly from the US samples. The citrus Liberibacter asiaticus has a really nice phage model in UF 506, which came out of Gainesville, I think in 2012. And ours doesn't really seem to match theirs, but we have more testing to do. But if you look at the New Zealand LSOA samples, they appear to have more phage diversity than our A. Liberibacter samples. And our LSO-B Liberibacter samples appear to have more phage diversity than our LSO-A samples. And then just recently, a paper came out on a new LAS genome, the Ishi1 genome. It's a citrus greening Liberibacter that came out of Japan that has no phage in it at all. And so looking at the Ishi1 genome, that these Liberibacter can exist, they can be pathogenic without phage, they can replicate, they can transmit. They ran the full gambit on Ishi1 strain LAS and could not differentiate it whatsoever from your typical Asiaticus. And then if you go and you look at Liberibacter crescens, that I think comes out of papaya, that doesn't appear to have any Liberibacter phage either, but that one is non-pathogenic. So when you look at Liberibacter and you look at some of our haplotype B samples that are missing phage entirely, the phage loss appears common, but yet the phage genes are conserved. And so we really don't have an explanation for that. That'll be something we'll be working on in the future.